Tyrannosaurus Rex, the heaviest terrestrial predator that ever walked the surface of this planet, one of the most extreme expressions of biological evolution that life on Earth has ever produced. An animal so terrifying, so perfectly engineered for destruction, that it has dominated human imagination for over a century. We've made movies about it, we've built museums around it, we've taught it to every child on the planet. We think we know it. But we don't, because in 2023, a team of researchers made a discovery inside a T-Rex fossil that should be scientifically impossible. Something that breaks the foundational rules of paleontology. Something that rewrites what we thought we understood about fossilization, about deep time, and about the most famous predator that ever lived. They found something that had no right to still exist after 66 million years. And once you understand what it is and what it means, you will never look at a dinosaur fossil the same way again. The impossible find. Here is what every paleontologist believed, without question, for over 150 years of fossil science. When an organism dies and becomes fossilized, its organic matter disappears. Completely, permanently. It's called diagenesis, a slow systematic chemical process where proteins, gnar, and every other biological molecule are destroyed and replaced by inorganic minerals. Bone becomes stone, chemistry becomes rock. The creature's biology is erased by geological time, leaving only the shape of what once lived. This wasn't a theory, it was the bedrock of paleontology. You could study a dinosaur skeleton, its structure, its proportions, its biomechanics, but the actual biochemistry of that animal, the blood, the proteins, the internal chemistry that made it alive, that was gone. Forever. Lost somewhere in 66 million years of deep time. That was the rule. And then scientists broke it. In the early 2000s, paleontologist Mary Schweitzer and her team at North Carolina State University were examining a T-Rex femur, a massive thigh bone recovered from Hell Creek Formation in Montana. The specimen was catalogued as Moore 1125. Standard procedure. Routine analysis. But when they dissolved the mineral matrix away from the bone's interior, something remained that should not have been there. Soft, pliable material. Transparent vessels. Structures that resembled blood vessels and bone cells. Not mineral casts of ancient tissues, not contamination, the actual remnant chemistry of the original organism. Inside a 68-million-year-old T-Rex bone, they found what appeared to be original biological tissue. The scientific community did not celebrate. It exploded in controversy. Critics immediately suggested contamination, modern bacteria, biofilm, anything to explain away a result that was simply too radical to accept. The idea that organic matter could persist across geological time at this scale violated everything the field had established. Extraordinary claims require extraordinary evidence. And the evidence at that point was not enough. So researchers went back. And they went deeper. What followed was nearly 2 decades of increasingly sophisticated analysis. New specimens, new techniques, new teams working independently to either confirm or destroy Schweitzer's findings. And here is what they confirmed. The proteins were real. The science. The technique that finally proved these proteins were genuine is called resonance Raman spectroscopy. And if that sounds complicated, here's what you actually need to understand. Scientists can use laser light to identify molecular structures without destroying the sample. They direct a 532 nanometer green laser at microscopic areas within fossil bone. When that laser energy strikes authentic hemoglobin molecules, it causes specific chemical bonds to vibrate at frequencies that are unique to hemoglobin and hemoglobin alone. It produces a molecular fingerprint. Not a guess, not an interpretation, a fingerprint. The iron-containing heme groups in hemoglobin, the component that carries oxygen in blood, produce a spectral signature that is chemically distinct from any known contamination source, from any mineral, from any bacterial byproduct. When the analysis was run on T-Rex fossil samples, that signature appeared. They were looking at fragments of actual dinosaur blood protein, preserved for 68 million years inside fossilized bone. But this immediately raises the obvious question. The question that every person watching this is already asking. How is that even possible? How does organic matter, notoriously fragile, notoriously susceptible to decay, survive for longer than the human species has existed by a factor of several thousand? The answer involves a mineral called goethite, an iron oxide compound that forms as hemoglobin degrades. As the original protein structure breaks down over geological time, the iron atoms from the heme groups don't simply disappear. They form protective mineral shells, microscopic chemical tombs, around surviving protein fragments. These shells create stable hybrid compounds, part organic, part mineral, resistant to the processes that would otherwise destroy them completely. Here is the staggering irony of this chemistry. The very process that destroys the protein is simultaneously preserving what's left of it. The iron acts as both executioner and embalmer. It breaks the protein down, and then it encases the fragments in a mineral shell that protects them from further degradation. In iron-rich burial environments, exactly the conditions found in Hell Creek Formation, this preservation process can maintain molecular signatures for timescales that, until recently, we believed were impossible. It's not contamination. It's not wishful thinking. It's chemistry that no one expected to find, confirmed by multiple independent research teams using different methods on different specimens. Now, before anyone gets excited about Jurassic Park, let me be very precise about what this does and does not mean. Hemoglobin fragments can survive through iron-mediated preservation. DINA cannot. The double helix structure that carries genetic information is far more fragile than blood proteins. It lacks the chemical properties that allow iron bonding at this scale. Complete genetic sequences cannot survive 68 million years, regardless of burial conditions. The fantasy of cloning dinosaurs from ancient Dinar remains exactly that, a fantasy. But what we can do is almost as extraordinary. What it means. Preserved blood proteins are not just curiosities. They are biological time capsules, chemical shadows of a living physiology that existed 68 million years ago. Before this discovery, everything scientists understood about dinosaur metabolism, physiology, and internal biology came from inference. You study the bone, you make assumptions about the animal that built it. Educated assumptions, carefully reasoned assumptions, but assumptions nonetheless. With preserved hemoglobin fragments, that changes. Researchers can now directly analyze the oxygen-carrying capacity of T-Rex blood. The molecular structure of hemoglobin reflects the metabolic demands of the organism, how efficiently it processed oxygen, how active its lifestyle was, how its physiology was adapted to the specific atmospheric and thermal conditions of the late Cretaceous. This is information encoded in the chemistry itself, not inferred from bone shape. For the first time in history, we have actual biochemical data from a living T-Rex, not a fossil impression. The original chemistry. The implications ripple outward into every major open question in dinosaur science. Was T-Rex warm-blooded or cold-blooded? The hemoglobin's molecular structure carries clues about metabolic rate. How did it manage oxygen at the scale of a 7-ton predator? The blood chemistry reflects the cardiovascular demands of that body. How did late Cretaceous atmospheric conditions, oxygen levels were slightly different 68 million years ago, affect its biology? The proteins hold that record. These are questions that paleontology has debated for decades using only bones. Now there is actual biochemical evidence to work with. But here is where this story takes a turn that almost no one anticipated. Because while scientists were processing the implications of preserved blood proteins, a completely separate line of research was simultaneously dismantling another century-old assumption about T-Rex. It wasn't hunting alone. The hidden predator. For over a century, the late Cretaceous ecosystem of North America had a simple predator hierarchy. At the top, T-Rex. Below it, everything else. The apex of apex predators, unchallenged, unrivaled, the undisputed ruler of the Cretaceous world. That story is wrong. The evidence has been sitting in museum collections for decades, dismissed and misidentified smaller Tyrannosaur specimens that paleontologists classified as juvenile T-Rex individuals. The logic seemed sound. You find a small Tyrannosaur, you assume it's a growing T-Rex that hadn't reached full size yet. Case closed. Except the anatomy didn't cooperate. These smaller specimens, collectively referred to as Nanotyrannus lancensis, possessed anatomical features that are incompatible with juvenile T-Rex development. Larger forelimbs relative to body size, a higher tooth count, fewer tail vertebrae, and most critically, distinct cranial nerve channel patterns, neurological pathways that are established early in development and do not change as an animal grows. Nanotyrannus had the wrong skull wiring to be a baby T-Rex. That doesn't change with age. You're born with it or you're not. But the debate persisted for decades because definitive proof required exceptional specimens. And in 2022, that proof arrived. The dueling dinosaurs. A fossil discovered in Montana's Hell Creek Formation in 2006 and finally made available for comprehensive scientific study after years of legal and acquisition disputes. The specimen contains a Nanotyrannus and a Triceratops preserved together, two animals locked in what appears to be their final confrontation, buried simultaneously in what was almost certainly an encounter that ended both of them. The preservation is exceptional by any standard, and it provided exactly the level of detail needed to settle the debate conclusively. Advanced analysis of the Nanotyrannus skull revealed mature cranial nerve development, not the underdeveloped pathways expected in a juvenile animal. Growth ring analysis of the bone tissue, the same method used to determine a tree's age from its rings, confirmed that this individual had reached skeletal maturity at approximately 20 years old. This was not a baby T-Rex. This was a fully grown adult of a completely separate species that had been misidentified for over 60 years. Further analysis revealed the situation was even more complex. Researchers identified not one, but two distinct Nanotyrannus species sharing the late Cretaceous ecosystem, Nanotyrannus lancensis, and a second species, Nanotyrannus rex. Each possesses unique cranial features, distinct dental arrangements, and postcranial adaptations that separate them from each other and from T-Rex. The name Rex for the second species references the river Lethe from Greek mythology, the river of forgetfulness, an acknowledgement of how long this animal went unrecognized, forgotten in plain sight inside museum collections around the world. 3 tyrannosaur species, sharing the same geographic range, at the same time, in the final million years before the end Cretaceous extinction. The Predator Guild. Think carefully about what 3 apex predators sharing the same territory actually means, because in modern ecosystems, this almost never happens at this scale. Lions and leopards in Africa avoid direct competition by partitioning their prey. Lions take the large animals, leopards take the medium-sized ones and retreat into trees. Tigers and leopards in Asia separate themselves through habitat preference. Even in the ocean, great white sharks, orcas, and tiger sharks occupy overlapping ranges only because the sea is vast enough to accommodate their different hunting strategies. But 3 tyrannosaurs, closely related animals, similar body plans, similar sensory systems, hunting the same Hell Creek landscape simultaneously? That requires a level of ecological specialization that paleontology had never previously attributed to non-avian dinosaurs. The size differences between the 3 species provide the first clue to how this worked. T-Rex, the largest, most likely focused on the massive armored herbivores of the period. Triceratops. Edmontosaurus. Animals that required 7 tons of predator to overwhelm. The 2 Nanotyrannus species, smaller and likely faster, probably specialized in medium-sized prey, animals that T-Rex's sheer bulk made awkward to pursue across variable terrain. This wasn't a simple monarchy with one king. This was a sophisticated predator guild, a system of ecological roles divided between closely related specialists. The behavioral implications are significant. In modern predator guilds, apex predators display complex social avoidance behaviors, territorial signaling, and prey partitioning that prevents direct competition. If 3 tyrannosaur species coexisted successfully for potentially millions of years, they almost certainly developed analogous behavioral strategies. That suggests a level of behavioral complexity in late Cretaceous tyrannosaurs that our previous models, built on the assumption of a single apex predator, never needed to account for. And this behavioral complexity connects directly back to the biochemical discoveries we started with. Because understanding how 3 apex predators coexisted requires understanding the neurological and sensory capabilities of each species. Which brings us to the biology that made T-Rex's dominance possible in the first place. The biological weapon. The popular image of T. Rex, as a lumbering, instinct-driven brute, has been dismantled by 3 decades of neurological research. What brain endocost studies, digital maps of the brain cavity's interior, reveal is something fundamentally different. T-Rex was a precision predator. Its dominance wasn't just size, it was sensory architecture. The visual system is the place to start. Unlike many large dinosaurs whose eyes were positioned on the sides of their skulls, providing wide field of view at the expense of depth perception, T-Rex evolved forward-facing eyes that generated significant binocular overlap. The result was stereoscopic vision, the ability to calculate precise distances with accuracy comparable to modern raptors. This is not trivial. Stereoscopic vision is metabolically expensive. Evolution doesn't produce it unless it provides a decisive survival advantage. For a predator that needed to time a bone-crushing bite attack with precision, too early and the prey escapes, too late and the strike is ineffective, accurate depth perception was the difference between eating and going hungry. T. Rex evolved its narrow snout not despite needing good vision, but specifically to maximize it. The skull shape is an adaptation for unobstructed forward sight lines. But the olfactory system is where T-Rex's biology becomes genuinely extraordinary. The olfactory bulbs, the brain structures dedicated to processing scent information, were proportionally massive in T-Rex. Larger, relative to total brain volume, than in virtually any other dinosaur species analysed through endocost study. The size of these structures indicates a chemical detection system of exceptional sophistication. Modern comparative anatomy gives us a frame of reference. Bloodhounds, the most olfactorily capable domestic dogs, possess roughly 300 million scent receptors compared to a human's 6 million. T-Rex's olfactory bulb proportions suggest capabilities that would have dwarfed even that. T. Rex could likely detect a carcass from multiple kilometers away, track a wounded animal across changing wind patterns, identify individual animals by their chemical signatures. This wasn't a nose, this was a biological radar system tuned to the chemistry of the Cretaceous world. The integration of these systems, stereoscopic targeting vision, extreme olfactory range, and the auditory capabilities suggested by its large cochlear structure, created a predatory package that operated on multiple simultaneous sensory channels. When one sense faced environmental limitations, the others compensated. Rain obscuring vision? The nose still works. Dense vegetation blocking sightlines? The ears still track movement. A prey animal that's hidden and silent? The scent still reaches. This is the architecture of an apex predator that had been refined by millions of years of evolutionary pressure into something very close to optimal for its environment, the bigger picture. Here is what you are left with when you pull all of this together. A predator we thought we knew, completely. A species that has been in museums and classrooms and popular culture for over a century. the most recognizable extinct animal in human history. And in the last decade alone, we've discovered that it had blood proteins still chemically intact inside its bones. That it shared its ecosystem with not one but two other tyrannosaur species that we had misidentified for 60 years. That its sensory biology was sophisticated in ways that our previous models completely underestimated. Everything you thought you knew about T-Rex was incomplete. And the most astonishing part is what this implies about everything else. If we were this wrong about the most studied, most scrutinized dinosaur in the history of paleontology, what are we still missing about the thousands of other species that haven't received that level of attention? The fossil record is not a finished document. It is an ongoing investigation. And with every new technology, resonance Raman spectroscopy, CT endocast mapping, advanced growth ring analysis, we get access to evidence that previous generations of scientists simply could not see. The preserved proteins in T-Rex bone are not just a biochemical curiosity. They are proof that deep time still has secrets. That the 66 million year wall we assumed separated us from the biology of the Mesozoic is more permeable than anyone believed. The past is not as gone as we thought. The work continues. New specimens are being analysed, new techniques are being developed, and the T-Rex, the animal we've known longer than almost any other, is still actively revealing things that change the way we understand life on this planet. Which means the story isn't over, it's just getting started.